Hello all, my name is O'Neill Mahoney. Welcome to my presentation. Accelerating the development of estrogenic predictive models utilizing large-scale databases. I would like to start off by thanking my mentors Lauren Wickstrom and Emilio Galicchio, my peers Valeria, Lynn, and Marwa, and finally, the CUNY Research Scholars Program and the iCompute program. Thank you all for your generous support. Estrogenic chemicals in the environment have generated public concerns over potential carcinogenic effects and other endocrine-dependent outcomes. Xenoestrogens are synthetic compounds that simulate the effect of estrogen and interfere with the process of the estrogen binding to the estrogen receptor. One well-known xenoestrogen is the BPA molecule, which has led to large campaigns geared at lowering its presence in our environment. Experimental studies have identified various xenoestrogens for their potential role in cancer development. However, these studies do not always focus on the structural models of the compounds within the binding process to the estrogen receptor. In addition, assays have demonstrated that many xenoestrogens bind weakly to the estrogen receptor in vitro, yet it is often unknown whether the compounds mimic the magnitude of the estrogen's response in vivo. Our goal is to address this gap in knowledge and learn more about endocrine disruption and risk through novel computational modeling techniques. My work in this project has focused on the construction of a chemical database. Our first database, the ligand database, stores information for approximately 200 potential chemicals that were further classified into 11 different groups. 3 examples of these classifications are presented. 17-beta-estradiol from the steroidal estrogen class, methylparaben from the parabens class, and DDT from the organochlorines class. You might recognize that name. DDT is one of the most famous compounds that was widely used and then eventually later identified to have an immense environmental impact. Presented is an example for the entry BPA. Also present are identifiers such as the IUPAC name, as well as physical properties such as the molar mass, and lastly, binding free energy calculations such as the delta G and IC50 values. Another of our databases, named the Protein Database, lists meta-information on 34 experimental structures sourced from the Protein Databank. Presented is an example of the estrogen receptor complex with estradiol. This data is essential for calibration of the work. Through comparison, we can ensure our emulation-based predictions reflect reality. The protein database has a weakness, however. Though we have approximately 200 compounds, only 14 unique structures were found to be resolved and present on the protein databank. This begs the question: can computational methods predict the experimental structure of a ligand in the ER binding pocket? Well, yes, it can. The glide docking program calculates binding free energies based on the best binding pose. The program uses experimentally tested algorithms to determine which compound interactions are most favorable. We are able to confirm the accuracy of our methods by comparing an experimental structure sourced from the PDB and one of our SMILE-generated structures. Here within the estrogen receptor, we can see a perfect match in the experimental pose in pink, juxtaposed with the predicted structure in blue. These databases we have created not only serve as an interface for analysis, but also as a growing brain. Once our database has an adequate quantity of data, we will use it in conjunction with Frontera, the world's fastest academic supercomputing network, to develop a predictive computational model for the binding of potential xenoestrogens to the estrogen receptor. Until then, our team will continue to gather data, growing our database and working to ensure repeatable and accurate calculations. Once again, thank you for coming to my presentation, and I hope you have a wonderful rest of your year.